Dear colleges, we, members of ISDC team, are happy to present to you a webinar developed within the framework of an ongoing grant project funded by the International Science and Technology Center. The project is titled Genetic Diversity of Mycobacterium Tuberculosis Complex Isolated from Agricultural Animals in Georgia. The webinar covers two main topics: a demonstration of the methodology for examining slaughtered animals, and identification of animals with tuberculosis symptoms. It is designed for veterinarians, veterinary students, sanitary epidemiological control specialists, slaughterhouse workers, farmers, and livestock industry professionals interested in identifying tuberculosis-related changes in slaughtered animals, collecting appropriate laboratory samples, and transporting them to the relevant laboratory for further diagnostics. In this webinar, we will cover the following topics: visual examination of the lymph nodes and lungs of slaughtered animals, proper sampling procedures, including collecting methods, storage protocols, packaging guidelines, and transportation of samples to the relevant laboratory for further research, proper methods for the disposal of infected carcasses, sanitary standards and requirements, the disinfection of equipment and facilities after working with infected animals, preventive measures to control the spread of tuberculosis in farm environments. Let's start by saying a few words about tuberculosis in general. Tuberculosis is an infectious zoonotic disease caused by several bacteria species within the Mycobacterium tuberculosis complex, (MTBC). You can see their list on the screen. It affects humans as well as various domestic and wild animal species. Agricultural animals, particularly cattle, play a significant role in human infection, especially in rural areas or among those working in the livestock industry due to humans can be infected through close contact with infected animals or by consuming contaminated animal products, such as milk, cheese, or meat. Among species in the list, Mycobacterium tuberculosis and Mycobacterium bovis are particularly noteworthy. It is established that Mycobacterium tuberculosis is the primary cause of tuberculosis in humans, while Mycobacterium bovis is the main pathogen in cattle. However, Mycobacterium bovis also plays a role in the development of extrapulmonary tuberculosis in humans, accounting for 12% of such cases. Pathogenesis. There are two main routes of infection in animals: alimentary and aerogenic. Once the pathogen enters the body, it disseminates via the bloodstream and lymphatic system. The bacteria then localize in various organs and lymph nodes, triggering an inflammatory response. This process leads to the formation of characteristic tuberculous granulomas, also known as tuberculomas. The image on this slide illustrates two key manifestations of tuberculosis: lymphadenitis, inflammation of lymph nodes; pulmonary tuberculomas, granulomatous lesions in the lungs. These pathological changes are hallmarks of the disease progression and are crucial for diagnosis and understanding the severity of the infection. In live animals, the diagnosis of tuberculosis is based on allergic tests, tuberculinization, and laboratory examinations such as microbiology, serology, and biomolecular analysis. Post-mortem diagnosis centers on detecting pathological changes in organs followed by laboratory confirmation. Key pathological changes to observe include nodular degeneration in tissues, commonly referred to as tuberculomas, alterations in lymph nodes, particularly mediastinal, bronchial, submandibular, retropharyngeal, and mesenteric lymph nodes. Organ damage rarely occurs without lymph node involvement. Here we can see the process of post-mortem examination of internal organs in slaughtered animals at a slaughterhouse. It is important to note that the examination of each animal is carried out in compliance with all safety protocols, even if there is no reason to suspect that it may pose a risk to others. This is the example of surface tuberculous nodules, tuberculomas, in lung tissue. During the examination, it is necessary to carefully palpate the investigated organs, as tuberculomas, identifiable as dense formations in tissue, may be easily missed during visual inspection, while palpation yields good results even if the disease has not progressed too far and the tuberculomas are not fully formed or not very dense. The slide shows us tuberculomas in the liver and heart. It is important to note that tuberculoma formation in the heart is an extremely rare occurrence. Here, you can see the mediastinal and bronchial lymph nodes of cattle. Due to the toxins produced by tuberculosis mycobacteria, caseous necrosis has developed in lymphoid tissue, surrounded by a fibrous capsule. In some cases, these caseous masses harden, leading to a condition known as calcification. This slide shows a mediastinal lymph node undergoing dystrophic necrotic changes with a distinct hyperemic zone, indicative of tuberculous infection. Upon detecting such changes in a lymph node, it is essential to carefully examine the organs associated with that node. These photos illustrate lung damage caused by tuberculosis. Based on lung anatomy, the following patterns of damage are distinguished. Acinar damage affects individual bronchioles. Acinar nodular damage, when affected acini coalesce. Lobular damage affects specific lobules of the lung. In lobular tuberculosis, entire lung lobes are affected, often leading to cavernous processes, tuberculosis-induced cavities in lung tissue. If such extensive lesions are detected, it is necessary to examine all organs of the animal, as a generalized form of tuberculosis is highly probable in this instance. Therefore, it is essential to strengthen safety measures. The next few slides will show tuberculosis-related changes in various internal organs. Current slide presents tuberculomas and calcification processes in bronchial and mediastinal lymph nodes. Here it is shown a tuberculoma in the liver, with caseous necrosis clearly visible in the left image. In cases of liver involvement, tuberculomas may not be externally noticeable, as they often lie deep within the organ. Additionally, the liver tissue itself is quite dense, and palpation may not guarantee adequate results. Therefore, during examination, it is necessary to make several incisions. This image illustrates a case of cardiac tuberculoma. It is important to note that tuberculoma formation in the heart is an extremely rare occurrence and typically arises as a result of the development of a generalized form of the disease. The slide displays spleen lesions, with extensive caseous deposits and surface tuberculomas. In cases of tuberculosis infection, this organ is not typically characterized by deep tissue lesions. Instead, all changes are generally superficial. The serous membrane becomes inflamed, while the spleen itself is enlarged and hardened. Here, we see severe lymphadenitis affecting serous membranes due to tuberculosis. Such lesions of the lymphatic system can be observed not only in cases of tuberculosis, but also in other infectious diseases. Upon their detection, it is necessary to examine carefully the adjacent lymph nodes to determine the specific underlying cause. This slide presents mesenteric lymph node tuberculosis. The extensive distribution of tuberculomas in the left image is sometimes referred to as a pearl-like form. This slide shows tuberculosis progression in the udder. Notably, alongside calcified tuberculomas, purulent masses rich in millions of mycobacteria are visible. If tuberculous lesions are detected in the udder, the risk of infection for potential consumers of milk and dairy products is very high. In such cases, it is necessary to screen the animal's owner and other at-risk individuals for extrapulmonary tuberculosis. This image presents bone tuberculosis. Recent research has confirmed the involvement of Mycobacterium microti in bone tuberculosis development. Generally, Mycobacterium microti is considered a tuberculosis pathogen in rodents, but modern studies have shown a strong pathogenicity of these bacteria in humans as well. The next slide depicts tuberculomas in the kidneys, with caseous necrosis clearly visible on the cross-section. As noted earlier, tuberculosis primarily affects the lungs in both humans and animals. To evaluate lung lesions, we utilized a 5-point scoring system proposed by Professor Natalie A. Parlane from Massey University. We assessed the apical, cardiac, and diaphragmatic lobes of the left lung and the apical, cardiac, diaphragmatic, and accessory lobes of the right lung. The slide provides a clear anatomical topography of lung lobes. Assessing lymph node damage is also crucial. We used Professor Natalie A. Parlane's 3-point scoring system. 0 points, no necrosis or visible lesions. 1 point, a single small lesion, 1 to 2 millimeters. 2 points, multiple small lesions, or a single large necrotic area, at least 5 by 5 millimeters. 3 points, extensive, total, necrosis. It is essential to evaluate all lymph nodes in the thoracic cavity, abdominal cavity, and head region. The slide illustrates the topography of mediastinal and tracheobronchial lymph nodes. It is also essential to evaluate all parenchymal organs and highlight all macromorphologically visible changes, including color, consistency, tissue tension, uniformity, and other characteristics. A crucial component of the post-mortem assessment process is filling out the animal's questionnaire. It is necessary to include the animal's individual number, the owner's personal identification number and contact details, breed, age, sex, geographical origin, and habitus on the left side of the slide. You can see the electronic form used by the United States Department of Agriculture for post-mortem animal evaluation. For better clarity, an explanatory section is attached to the form, detailing how it should be filled out. All links from the slides are available in the webinar description below the video. When macromorphological changes resembling tuberculosis are detected in slaughtered animals, it becomes essential to collect samples for further micromorphological and laboratory examinations using various methods. For micromorphological analysis, it is important to collect pathological material correctly, considering the following. Samples should not be taken from calcified tuberculomas, as microscopic examination of such tissue will only show a homogeneous, structureless image. Samples should be collected from the border of necrotic and less damaged areas of the tissue. Regarding sample collection for microbiological examination, priority should be given to collecting material from affected lymph nodes, affected parenchymal organs. In generalized forms, it is necessary to collect samples not only from damaged tissues, but also from all adjacent lymph nodes. Before sending the samples to the laboratory, they must be processed and preserved as much as possible. Fatty tissue should be removed. For histological examination, it is preferable to place the material in formalin. For microbiological examination, if delivery to the laboratory is possible within 48 hours, the sample should be stored at 4 degrees Celsius. Otherwise, it is best to freeze it at -20 degrees Celsius. For long-term storage exceeding 1 year, the material should be frozen at -80 degrees Celsius or lower. The same requirements apply to biomolecular studies. The packaging, labeling, and transportation of samples are regulated by specific guidelines. Each country has its own regulations, but all adhere to the recommendations provided by the World Health Organization, WHO. The slide presents a guide titled Guide to Infectious Substance Transport Regulations. Regarding air transport, it is regulated by the International Air Transport Association, IATA, and categorized into 2 groups, A, UN 2814/UN 2900, and B, UN 3373. Category A includes biological materials that pose a danger to both humans and animals, specifically referring to live biological agents. Unlike Category A, Category B pertains to diagnostic samples, including human or animal specimens intended for clinical testing, research, or disease diagnosis. Category B does not contain materials that cause human or animal diseases, WHO has developed specific regulations and guidelines for sample packaging. The slide displays the packaging scheme for Category A and B shipments. In both cases, the use of an absorbent and double packaging is mandatory. For Category A, the secondary packaging must also be waterproof, often utilizing metal containers. Specific labeling is essential, and for international shipments, it is regulated by the aforementioned guidelines. The label must include the animal's identification number, allowing for detailed information retrieval. Additionally, it must indicate the sample category. In our research, we included additional information on the label, such as breed, sex, age, region, and the slaughterhouse where the animal was processed. Each lymph node and affected organ was individually packaged and labeled to facilitate subsequent scientific analysis. The next topic to address is the proper disposal methods of carcasses. The gold standard is incineration in specialized furnaces, incinerators, where the temperature must exceed 850 degrees C. it is crucial to conduct the burning process away from public areas and ensure the presence of authorized and trained personnel. Another recommended method, endorsed by the World Organization for Animal Health, is alkaline hydrolysis. This method uses a pressurized chamber to dissolve carcasses in an alkaline solution. When using this method, special attention must be paid to preventing wastewater contamination, and the process must be carried out by licensed operators. Another method of disposal is burial, which is used when incineration or alkaline hydrolysis is not an option. Burial must occur only in designated landfill sites and requires special permits from environmental protection authorities. The most critical factor in this method is preventing biological fluid leakage from the burial site and groundwater contamination. In many cases, burial sites are lined with protective, liquid impermeable materials. The burial depth must be at least 2 meters and covered with an additional dense soil layer to prevent scavengers from digging up the remains. As mentioned, carcass disposal is regulated by national legislative directives and guidelines developed by the World Organization for Animal Health. One of the most critical aspects of this process is personal safety. Personnel must use disposable coveralls, gloves, face shields, N95 respirators, and protective eyewear. All instruments, equipment, and transport vehicles must undergo disinfection with disinfectant solutions effective against tuberculosis bacilli. During disinfection, special attention must be paid to preventing wastewater contamination. The following legal requirements must be met: proper documentation of carcass transportation, disposal method, and safety measures. Environmental protection agencies have specific reporting forms. Detailed information about the disposal process must be promptly reported to epidemiological surveillance authorities to ensure appropriate preventive and safety measures are implemented. The disposal process must be inspected and audited by relevant epidemiological surveillance agencies. Although we frequently mention the disinfection process during our webinar, it is necessary to discuss this process in more detail. As previously mentioned, tuberculosis in agricultural animals is caused by mycobacteria from the Mycobacterium tuberculosis complex, most commonly Mycobacterium bovis. This microorganism is resistant to both alkaline and acidic environments and is resistant to many disinfectants. However, certain disinfectants, when used correctly, ensure its elimination. Regulations and recommendations regarding the disinfection of tuberculosis-causing mycobacteria have been developed both by the World Organization for Animal Health and local regulatory authorities at the legislative level. In the webinar, we briefly highlighted the use of some disinfectants. It is important to note that most of these substances are toxic and require strict safety measures when used. Alkalize a 2% solution of sodium hydroxide with a 30-minute exposure is effective in eliminating all mycobacteria within the Mycobacterium tuberculosis complex. Phenolic compounds, a 0.5% aqueous solution of phenol with a 30-minute exposure time, is effective. In this case, biosecurity measures must also be followed. Peracetic acid, a 0.2 to 0.5% solution with an exposure time of 10 to 15 minutes. Highly toxic, requiring gloves and goggles. It has a corrosive effect, so it is not used for disinfecting metal surfaces. Sodium hypochlorite, a 0.5 to 1% solution kills tuberculosis bacilli within 30 minutes. Sodium hypochlorite solutions are widely available in household chemical stores under various brand names, commonly known as bleach. When preparing the solution, safety precautions must be taken, as it is toxic and a strong oxidizer. The next topic in our webinar addresses preventive measures in farms to prevent the introduction and spread of tuberculosis. Key preventive measures include: 1. Screening and quarantining newly introduced animals. TB testing must be conducted on all new animals before they are introduced to the farm. This typically includes tuberculin testing or, where possible, serological tests such as the gamma interferon test. Before introduction, animals must be quarantined for at least 30 days, during which they should be observed for clinical signs of disease, and if necessary, retested for tuberculosis. A qualified veterinarian must examine new animals for TB or other infectious diseases before they mix with the existing herd. Another important component of tuberculosis prevention is controlling farm boundaries and entry points. Secure fencing. The farm should have sturdy, double-layered fences to prevent direct contact between farm animals and potentially infected wild or stray animals, such as wild cattle or stray dogs, which may carry Mycobacterium bovis. Controlled access points. Entry to the farm should be restricted, allowing only essential personnel, vehicles, and animals. Strict biosecurity measures should be in place for visitors, including designated clothing and footwear. Signage and biosecurity zones. Clearly marked biosecurity zones should restrict access to critical areas such as feed storage and animal shelters, with access limited to authorized personnel only. Regular testing and monitoring of animals. Regular TB testing of animals should be conducted annually or biennially to detect infections early and prevent disease spread. TB cases should be monitored in local animal populations in collaboration with veterinary authorities. In regions where TB is endemic in wildlife, additional preventive measures, such as special fencing, should be implemented to minimize contact. Accurate records of TB testing and monitoring results should be maintained, and management of animals and equipment. Regular refresher training should reinforce these principles. Hygiene and sanitation standards in farms. All common equipment, such as feeding troughs and milking machines, should be regularly cleaned and disinfected with effective agents such as phenol and chlorine-based disinfectants. Disinfection points, so-called disinfection barriers, should be installed at the entrance to animal enclosures. Waste management must comply with biosecurity norms. Preferably, biological waste should be processed to produce biofuel. Waste should be securely stored to prevent access by wild animals, which could be potential disease carriers. Ensuring the safety of animal feed and drinking water. Diseased animals must not come into contact with feed and water sources. These should be stored separately, away from wild animals and rodents. If animals drink from natural sources like lakes or rivers, it is crucial to ensure that the water is safe and free from contamination by wild animal excrement. In some cases, veterinarians may recommend dietary supplements with immunostimulatory or radioprotective properties, but their use should be supervised by a veterinarian. Training and health monitoring of farm personnel. Biosecurity training. All farm personnel should receive training on biosecurity practices, including proper hygiene, disease recognition, and management of animals and equipment. Regular refresher training should reinforce these principles. Health monitoring of workers. Workers in direct contact with animals should undergo health monitoring, as humans can also carry TB. Anyone showing signs of TB infection should seek medical attention and avoid contact with animals until they recover. Personal protective equipment, PPE. Workers must use protective gear, including gloves, masks, and special footwear, especially when handling animals or cleaning farm facilities. Wildlife as tuberculosis reservoirs. Control and monitoring. Cooperation with local authorities is essential to monitor and manage wildlife populations known to carry TB, such as wild cattle, stray dogs, wild boars, and badgers. Food, animal products, waste, and other materials that attract wildlife should be stored securely to prevent access. TS should be maintained, and any detected cases must be promptly reported to state authorities for appropriate quarantine and disease control actions. Vaccination as a preventive measure, where applicable. Some regions allow the use of experimental TB vaccines for cattle in high-risk areas under strict veterinary supervision. However, vaccination must be carefully managed, as it can interfere with TB testing. Some countries implement wildlife vaccination programs, e.g., oral vaccination of badgers in the UK, which help reduce TB transmission among wildlife and indirectly protect farm animals. Participating in or supporting such programs may lower local TB risks. Regulatory aspects of tuberculosis control in Georgia. The regulation of animal health, including the prevention and control of tuberculosis, TB, in livestock, is governed by several national laws and regulations. Key acts related to animal health, biosecurity, and disease control include those listed on the slide. All these regulations align with the guidelines and recommendations set forth by the World Organization for Animal Health. Thank you very much for your attention.